Okay. Okay, so happy Halloween to everybody. And we have a good topic for Halloween today because we're going to get into some of the oddities of fungi, which definitely qualify as great organisms for thinking about the kinds of things we typically think about on Halloween, like the supernatural with their strange chemical properties, as well as things that are pretty creepy and even hideous, which some fungi definitely demonstrate. So I'm not going to show you all the images I could to get to the extreme end of that spectrum, but I'll talk about it a bit. So we're going to focus, this will be the last lecture on fungi, and we'll continue to look at some of the major groups and things that have been traditionally considered fungi but turn out not to be, based on recent evidence that are pretty cool organisms. And definitely you'll see one of these, actually a couple in lab if you haven't already. And we'll look at a characteristic fungal life cycle, which is about as different from our life cycle as any group of organisms. And finally, we'll look at some ecological and human importance of fungi, which is the most interesting in some ways. And then we'll start to look at photosynthetic organisms in detail today. Actually, we'll just start to, I'll give you a broad overview if we have time, and then we'll get into that a little bit more next time. We're going to particularly look at the origins of photosynthetic capability, which in a way is also a kind of interesting Halloween topic too. So I mentioned last time that most macrofungi that we see out in the environment, we're only seeing the reproductive structures, and the bulk of the mycelium or fungal body is out of sight. It's penetrating its food source, and so we're not able to see it. And back in the early 90s in Michigan, some researchers were studying some honey mushrooms, which are in the genus Armillaria, and they started to get confused because they were genotyping mushrooms of Armillaria across a particular area, trying to look at population biology, and they could only find one genotype. The more they looked, the more they just saw the same genotype from molecular data. Finally, they realized they were sampling a single individual of this species that extended over about 37 acres. And so this, based on their calculations, this organism was larger than, in terms of biomass, larger than a blue whale, hundreds of tons in biomass, and probably based on the rate of growth of these things, probably around 1900 years old. So ancient organism. And the local folks capitalized on this for tourism and started their Humongous Fungus Festival, which still goes on. I actually found online there was the 21st annual one happened this August. But as the quote indicates up there, people generally are not too happy to find out that there's no gigantic mushroom the size of a giant redwood. When they go there, it's all hidden underground. And even worse, recently there have been larger honey mushrooms found in the Pacific Northwest of different species that have been estimated to be almost 9,000 years old, which would make it older than the oldest known plant, oldest known living thing, the bristlecone pine that's found in the desert mountains of the west here. So these things can get to be pretty huge and pretty interesting. So that's Halloween story number one in terms of thinking about the giant fungus under your feet growing away there. The last major group of fungi I want to talk about, I didn't mention this one in that phylogeny, and the good reason for that is that this isn't a phylogenetic grouping, and it's really more of an ecological grouping now. It used to be it is still considered a taxonomic group, the Duromycota, which means the second fungi, sort of the second tier of fungi, because this is basically the taxonomic garbage can that mycologists put fungal species in when they can't figure out what group they belong to. And the reason that they haven't been able to figure these out traditionally was that they don't produce any sexual structures, and the sexual structures are the ones that are most diagnostic for these different major clades or phyla of fungi. They've also been called the fungi imperfecti, or the imperfect fungi. And in botany, imperfect doesn't mean necessarily flawed in some fatal way. It just means that something's missing. And in this case, it's the ability, or at least the evidence of any sexual reproduction. Most of them, about 25% of all fungal species have been classified in this group. So there are a lot of fungi for which no sexual reproduction is documented. But molecular data have allowed us to place these things now in almost the vast majority of them in one of those 5 major clades. And a lot of them are ascomycetes or basidiomycetes. Actually, if you look at the presence or absence of septi within the hyphae, you can figure out whether it's a higher fungus or not just from that alone. And these are mostly have septate hyphae, so ascomycetes and basidiomycetes. And it's kept as a group for ecological purposes just because asexual fungi have some features in common that are worth considering by ecologists. All right, so now we're going to leave the major groups of fungi, and we're going to focus on a group that has a name that suggests it's a fungus. The ending mycota refers to fungus, but these are, and they have a common name, water molds, that would suggest that they're fungi as well. And if you look at their bodies, as you can see from the hyphae radiating out from this dead cockroach, it's also kind of a Halloween image there too. You can see that it looks pretty fungus-like. But if you actually look at the ploidy of the nuclei inside of those hyphae, they're diploid, like our nuclei in our mature bodies. The only haploid end stage in these water molds are their gametes, so it's just like us, has a life cycle like us, a gametic life cycle. The fungi, though, instead, as I'll mention in more detail in a moment, the only diploid stage for them is the zygote. So it's almost the opposite extreme. So they really have a different life cycle than fungi. And also their gametes, if you look at their gametes look a lot like ours, not at all like fungal gametes. They have morphologically different gametes. They have sperm and they have eggs. Sperm that are small and motile with flagellae, and eggs that are larger and non-motile. So that's the basis for the name, the egg fungi, which is an old name, which is literally a translation of mycota, the egg fungi, because other fungi true fungi don't have eggs and sperm. And there's a good reason why these were an exception, because it turns out they're actually more closely related to some of the algal groups we're going to talk about later, like the brown algae and the diatoms, than they are to fungi or animals. Okay, this is a group that includes important decomposers, but it also includes some really notable disease organisms, and especially of plants. One of the most notable outbreaks of disease caused by a member of this group, these disease organisms are mostly in the genus Phytophthora if you're interested, was the Irish potato famine back in the mid-1800s that resulted in the death of about 1 million people and the displacement of about 1 million more because the organism was pretty destructive to the potato plants. And Phytophthora species have a long history of being inadvertently, accidentally introduced into different parts of the world where they aren't native. And when they do, they often cause havoc to the native flora or to agricultural plants. And here in Western North America, we have some major diseases that have been around for a couple of centuries, for well over 100 years. The Port Orford cedar blight in the Pacific Northwest has wiped out much of the Port Orford cedar stands. And more recently, and possibly more seriously, in the last 25 years, we've had the outbreak of a disease called sudden oak death here in California that's killed tens of thousands of oak trees. And even worse, it threatens the extinction of our native tan oaks, which is a really important food source for a lot of wildlife and our major constituent of our mixed evergreen forests in coastal California. So over on the right, you can see some dying oak trees there in Marin County where the disease was first recognized. And it's pretty clearly an introduction from some other part of the world, although that hasn't been established as to where it's from yet. There's basically one clone. There are two clones known, but one of them is the one that's invaded California, and it's spread widely. So the fact that these things can get around pretty easily, they do have motile spores as well that can swim, and they can swim from tree to tree. They can be transported sometimes only through the soil, sometimes through the air as well. And unfortunately, the sudden oak death pathogen, Phytophthora ramorum, can get around in air and soil. So it's a really important group ecologically, in part for unfortunate reasons. All right, so one of the coolest of all the groups we're going to talk about, but unfortunately just briefly, are the cute and cuddly slime molds, which are one of the strangest groups of organisms in the world. And you'll see a plasmodial slime mold in lab if you haven't already. But these are organisms that are they look something in between an animal and a fungus. They're in a group called the amoebozoans, which literally means the amoeba-like animals. And you could argue that they have the largest cells in the world because this particular group of slime molds that we're going to focus on here, the plasmodial slime molds, they don't form any compartments around their nuclei. The spores germinate. They form unicellular amoeba-like organisms that move around by pseudopods, just like a true amoeba. If you've seen the old movie The Blob, similarly moving their limbs of cytoplasm around and engulfing small prey items. So they're carnivores, or at least predators, I should say, going after bacteria, fungal spores, small particles. They eat a lot of detritus. They're often found on forest floors, moving around on dead logs or dead leaves. And they can basically move around, finding new food sources and engulfing them. Actually, the original the body that germinates from them functions as a gamete. The individual little amoebas will fuse together different mating types and then form a diploid organism. And a zygote at that point that then becomes a diploid organism that just keeps growing. You can see on the right there how they ramify and get to be bigger and bigger, spreading out over their environment to find food. They're often really brightly colored and interesting. And some people are real aficionados of these things and keep them as pets. They can grow up to about 20 centimeters in diameter. And the mycology professor that I took general biology from many years ago, he kept these things and studied them in his office, and he fed them ground up wheat thins and Triscuits, and they enjoyed that a lot, and they grew. But once they run out of food, or they can't, the weather changes in a way that's detrimental to them, dries out or gets too cold, they'll start to solidify and produce a sporangium, as you can see over here on the left, within which meiosis will occur and produce myospores. So these are really interesting organisms that are often neglected, but they're all around. You can find them in the local woodlands if you look carefully, and they are fairly brightly colored, so it's not that hard to pick them out. They undergo an interesting process called cytoplasmic streaming. So if you wonder about how an organism that effectively is like about a 10 to 20 centimeter wide cell, full of up to like 1,000 nuclei or more, thousands of nuclei, how can it actually distribute cytoplasm around across that whole body? Well, it's capable of moving cytoplasm back and forth, and you can see that in this little video up here in the upper right. Might be a little bit hard to see. It'll take off here in a second. There, you can start to see some particles moving around there. Might be a little hard to see from the back of the room, but they do have cytoplasmic flow that's at a high enough rate to where you can see it with the naked eye. You don't have to use time lapse. All right, so those are slime molds. All right, so and the slime molds, just like those Mycota, used to be thought to be fungi, but it turns out that their relationship is no more close to the fungi than it is to animals. So this amoebozoan group that they belong to is sister to the clade that includes the animals and the fungi, and they split off before the animals and fungi split off from a common ancestor. So maybe not surprising that they have some semi-animal-like and fungal-like characteristics. And this, so let's move on now. We really need to talk about life cycles in order to understand the biology of most of the organisms we're going to talk about in the course. And although no doubt many of you consider yourselves experts in sex, when it really boils down to it, sexual reproduction across all organisms has a couple of things in common that are really fundamental and are commonly overlooked. So it's important that we review this. And more than any other thing in this part of the course, you really have to take this part to heart because we're going to be looking at life cycles of lots of organisms going forward. And if you can't place them in the overall general life cycle of sexual life cycle, it's going to make it hard to compare these groups to one another. And in order to understand how they evolved and the significance of all that, you really have to understand sexual life cycles. So fundamentally, sex involves two processes, and this is shared across all sexual organisms. And these are meiosis and fertilization. Fertilization is also sometimes called syngamy, or the fusion of gametes. Syn refers to fusion, or united actually, and gamete refers to the gametes, like sperm and egg. So sexual reproduction occurs widely across the natural world, and it's thought to be crucial for producing the kind of variation necessary for organisms to be able to respond to environmental challenges. So both of these different processes, meiosis and fertilization, are sources of genetic variation. Of course, mutation ultimately is the source of genetic variation, but mutation rates are incredibly slow, and most mutations turn out to be deleterious or harmful. But basically, making sure that variation is widely distributed within populations is a major factor here with regard to meiosis and fertilization. So the meiotic process is the one that's least familiar with to most people. And meiosis is often referred to as just a type of cell division, one that results in the reduction of the diploid or two-end condition to the one-end condition prior to fertilization, prior to gametes being formed and fertilization occurring. But meiosis is also a source. Fertilization is obviously a source of genetic variation. You're bringing together gametes from different individuals that have different genetic constitution to make a new individual of unique genetic constitution. But meiosis contributes importantly to this process too. So in your own germ cells, in your gonads, there you have, like in all of your other diploid cells, you have one set of chromosomes from your father and one set of chromosomes from your mother. And those chromosomes, they look like the ones in your mother and father. Well, actually, those chromosomes are similar to the ones that were generated by your parents in the gametes that they contributed that formed you. They're identical to the chromosomes that were in the gametes that formed you, is the best way to put that. But during the process of meiosis, you have the pairing of the chromosomes of your two parents. For each one of the sets of pairs of chromosomes in your each of the 23 pairs of chromosomes, the two chromosomes come together, pair, and we have genetic exchange, at least one crossing over event between those chromosomes that results then in a unique chromosomal state. So in order for the chromosomes to even be able to divide at meiosis in the first meiotic division, they have to have paired and undergone a crossing over event. And that crossing over event then yields a unique chromosome that combines elements of your parents' chromosomes together. So that's the importance of meiosis. I mean, it incidentally also reduces the ploidy down to haploid state, but the most important thing is that it's a source of genetic variation there. Okay, so you get recombined chromosomes out of that process, and you end up with haploid state in the resultant cells, of which there are four. There are four products of meiosis, not two, and that's because prior to the beginning of cell division, each of your parents' chromosomes undergoes duplications, resulting in two sister chromatids of identical constitution. And then you have 2 rounds of cell division, giving you 4 products, 4 different haploid cells, which in us immediately become gametes, but that's not the case for a lot of botanical organisms. All right, so are there any questions about meiosis and fertilization at this point? All right, so let's go on now to look at some life cycles. These are the 3 sort of generalized life cycles of different organisms on Earth. And these also appear in your textbook, so you can review them there. But all of these 3 life cycles share the feature that we have meiosis, followed by fertilization, followed by meiosis, followed by fertilization. It's a cycle of that sort. But how they differ is in the relative timing of meiosis and fertilization. So in us and other animals here on the left, meiosis is immediately followed by fertilization. So the only haploid stage in animals are the gametes, the sperm and the eggs. And of course, a gamete, unlike a spore, is a reproductive cell that fuses with another gamete, another reproductive cell to form a zygote that ultimately become a new organism. And it becomes a new organism, to put things in a really super simplistic way, because we have a lot of mitotic divisions following the zygote. These mitotic divisions, now mitosis, in contrast with meiosis, they're both types of cell division, but mitosis is resulting in an identical clone of the, you end up with 2 cells of identical genetic constitution to the original cell. So it's basically cloning or a replication giving identical cells of identical genetic constitution in most cases, all that we're going to worry about at least. And that will then ultimately give rise to an adult individual. So that's the animal life cycle. The fungi are all the way over at the right end of the spectrum. They're at the opposite end. And there we also have meiosis followed by fertilization followed by meiosis. But in this case, meiosis immediately follows fertilization. So the only diploid stage is the zygote. And immediately after the zygote is formed, we have meiosis that gives rise to 4 haploid spores that then germinate directly. Remember, spores germinate directly to form a new organism. So we have a lot of mitotic divisions giving us a new organism of haploid state. And eventually we'll have gametes formed from that organism, but gametes will then be formed by mitosis. The gametes will be genetically identical to the parent because it's not meiosis, they're already haploid organisms. It has to be mitosis. And then gametes, of course, will fuse from different individuals to give us a new organism, well, actually to give us a zygote that will undergo meiosis to form 4 spores that will each individually germinate to form new organisms. So it's a relative timing of meiosis and fertilization. It's also a second point here, whether mitosis is limited to one of those stages, and if so, which one, the haploid or the diploid stage. So in the case of animals, it's the diploid stage where mitosis occurs. In the case of fungi, it's the haploid stage where mitosis occurs. Or does mitosis occur in both stages, in which case we would have haploid organisms alternating in the life cycle with diploid organisms, which is a completely alien concept to an animal like us, but it's what it's basically what all the higher plants do, and some other photosynthetic organisms too. And we'll get back to that more later. All right, so this is just a very brief introduction, but this is really important to take to heart. So please, as soon as possible, study this. Totally, take this, make this second nature to consider. Draw this out on a piece of paper, quiz each other about it, whatever it takes. But you really do want to think about this and make sure you understand it, and try not to fall into the trap of constantly trying to interpret these life cycles in terms of our own biology, because we've just seen the 2 organisms, the slime molds and the Mycota, that have life cycles like us, and all the other botanical organisms we're going to look at are not like us. They're like us in the sense we have meiosis followed by fertilization, by meiosis, etc. But in these other respects, we're going to be looking at B and C, not A over here on the left. All right, so the fungi have one more wrinkle that you need to take into account that's pretty strange. I mean, most people, I think, coming into the class would think, well, I mean, maybe I wasn't sure about it. I'm not quite up to date on the details of meiosis, but I'm pretty sure I understand fertilization. And fertilization is a pretty straightforward process, fusion of gametes. That's not hard to grasp, but in fungi, it's a little bit harder to grasp. It's a little odd because in the higher fungi, the ascomycetes and the basidiomycetes, the fusion of the cytoplasm of the gametes, which occurs when sperm and egg come together in the case of animals,